Hi there, this is Josh Sommer, co-founder and executive director of the Chordoma Foundation. Thank you so much for joining this Impact Update webinar. It was originally recorded on May 12th, 2020. And our goals for this webinar were first to provide an update on some encouraging recent developments over the last quarter, to talk about how we're continuing to forge ahead in our mission in the midst of COVID-19, and also to provide an update on current priorities looking forward. I presented on these topics for about the first hour and then was joined for a live Q&A with our Director of Patient Services, Shannon Lozinsky, and our Director of Research, Joan Levy, where we took questions live from webinar participants. Of course, things may have changed since then, so for the latest, please check our website at chordoma.org, or feel free to get in touch with me directly at josh@chordoma.org. Again, thanks so much for joining. I'm going to dive into a review of our strategic priorities. For those who have been involved with the foundation for a while, I think you're probably familiar with our vision, but I think it's always important to focus on that first and foremost, and to let that guide us. And so I want to just reiterate that our vision is for everyone affected by chordoma to be able to overcome the disease and maintain their quality of life. And I think it's important to emphasize the last part. It's not just about overcoming the disease at all costs. It's about overcoming the disease and maintaining one's quality of life. And that means getting back to the life we love and getting on with our lives in a way that is wonderful and doesn't hold us back. So how do we achieve this vision? We have a two-pronged mission to improve the lives of those affected by chordoma and to lead the search for a cure. And ultimately, what's required to do that is to find better treatments, to deliver better care to patients, and to provide patients with a better experience. And when we talk about better treatments, it's important to emphasize here, these are treatments that enable patients, again, not only to live longer, but to live better. Treatments that are less invasive, treatments that ideally prevent recurrence, and if recurrence does occur, treatments that are able to control and ultimately cure patients who have recurrent or even metastatic disease. From a better care perspective, this is enabling earlier and more accurate diagnosis. It's ensuring that wherever patients are seen, that they're ultimately referred to specialists who have experience treating chordoma. It's ensuring that those specialists provide evidence-based best practice care. And from a better experience standpoint, this is ensuring that patients have the information, the guidance, and the support that they need to take on this disease confidently and feeling well informed and well supported, and able to access the care that they need with minimal barriers. What we recognize is that there are a series of programs that are required to achieve these goals: research to achieve better treatments, healthcare improvement to achieve better care, patient services to promote a better patient experience. And progress in each of these areas is mutually reinforcing. They don't stand alone in a vacuum. Progress in each of them is essential to the overall success of our mission. So over the last decade plus since the foundation started, we've made significant progress in each of these areas. Again, I've talked about this in more length in previous presentations, so I won't go into great detail, but I'll just highlight some key milestones and key achievements in each area. Over the last 12 years, we've, as an organization, invested now over $10 million in research. We've created a critical set of enabling infrastructure to make high-impact research possible. I think as many of you have been involved for a while know, when we started, there was really nothing for researchers to work with. We had no cell lines, we had no tumor tissue, we had no animal models, we had no way for researchers to access these critical materials. And a huge accomplishment over the last decade plus is putting in place those centralized repositories so that any researcher anywhere in the world can easily access the materials and the information that they need to study this disease. The combination of those things, creating that infrastructure, investing in research, and a lot of proactive outreach and networking has enabled us to attract and build a community of over 350 researchers from academic institutions across the world, as well as more than a dozen companies. This is all culminated in over 50 peer-reviewed publications. The output of that has been really vast, but a couple of key things to highlight: over 20 potential therapeutic targets have been identified in the last dozen years. And really importantly, something to underline is that there has been an accumulation of evidence, a real pile of evidence now that is built up, pointing to chordoma having an Achilles heel, essentially a unique vulnerability that it cannot live without, called brachyury, which I'll talk more about later. And that has posed some really tremendous opportunities. At the same time, and perhaps most importantly, this is all culminated in the Chordoma Foundation supporting 7 new clinical trials, and there have additionally been a couple others that have started. So now over 10 clinical trials that have started in the last dozen years, whereas previously there was only 1 clinical trial that had started when the foundation came into existence. On the healthcare improvement front, we've built a network of over 500 treating physicians across the world. This is in countries throughout North America, throughout Europe, and increasingly throughout Asia as well. In working in partnership with colleagues in Milan, we built a over 60-member global consensus group to try to generate a consensus around what are the best practices for treating this disease. There had been a lot of variation in terms of how it was treated. Patients would be treated one way in one institution, another way in another institution. That led to really variable and in some cases suboptimal patient outcomes. And so we wanted to make sure that there was a central source of truth for what were the evidence-based best practices for treating this disease. We've had 3 consensus group meetings in Milan, co-hosted with the Istituto dei Tumori, as well as the European Society for Medical Oncology. And the output of these was the first 2 treatment guidelines for managing chordoma, one focused on primary disease, one focused on recurrence. And I'm very happy to say that, in fact, the 3rd paper was just accepted, not yet published, but will come out in the next couple months, focused on how to prevent recurrence by adequately addressing the margins of tumors. So very excited that that's upcoming. From the state patient services standpoint, we've, as many of you have experienced, created a whole suite of services and resources to help provide education, navigation, and support. And over the last 4 or 5 years, under the leadership of our Director of Patient Services, Shannon Lozinsky, we've created a patient navigation service, which has provided one-on-one assistance to over 2,000 families across the world. We've created a doctor directory, a clinical trials directory. Many of you have benefited from these resources, all of which are available on our website. I'm very excited about the online community that we launched a couple years ago, Chordoma Connections. So in total, between our patient navigation service, connections, our events, et cetera, we've now served over 7,800 patients and family members from all over the world. So while it is the case that chordoma is a rare disease, and it is the case that for a long time it felt like we were, many of us were going this alone, that is no longer the case. We are in fact, when everyone is connected, quite a significant and strong community of 7,800 and growing. So I think if we take a step back and reflect, where has all this brought us? There's a lot of progress that's been made, but there have been a few really key milestones, a few really key turning points that have come together to create, for the first time, a credible path to treatments that not only could benefit patients in the future, but actually have a chance of benefiting patients who are alive today. And we've talked about some of those, but to highlight them, for the first time now, we have line of sight to the first drugs that can actually directly target brachyury, the Achilles heel of chordoma. we have quite a number of clues coming out of labs and out of clinics across the world that in fact there are better treatments that may already exist, some of which are in clinical trials, some of which will be entering clinical trials in the next couple of years. We've created the conditions for rapid progress by virtue of having created this research enabling infrastructure that I described and the research network that I described. Because we've learned so much about the biology of the disease, we are much closer now than we were several years ago to actually being able to not just study and understand the disease, but actually intervene and treat it. And then importantly, we have a united patient community, a united physician community, and united research community that is really key for making rapid progress in a disease like chordoma. So I think it's important to note here that having an opportunity like that, having an opportunity to impact the lives of individuals who are affected by a disease in the near term is really an opportunity that doesn't come along often in the world of oncology. It's something that is relatively rare, and it's really an extraordinary opportunity for those of us who are dealing with or have been affected by chordoma. And it's an opportunity that we as an organization and as a community, we know we must seize. And so to that end, we have launched our All In campaign, which many of you are aware of. We believe that if ever there was a moment to go big, if ever there was a moment to really swing for the fences, if ever there was a moment to go all in, this is it. And this is our rallying cry. We're aiming to raise $16 million to advance research, to promote healthcare improvement, and to advance patient services. And as a part of this campaign, we see 5 key areas of opportunity and 5 key corresponding campaign priorities. So first and foremost, perhaps the biggest opportunity over the long run to change the course of this disease is to discover drugs against brachyury. Secondly, to identify immunotherapies that could be effective for chordoma. There are many immunotherapies that are being developed and billions of dollars that's being invested to create new immunotherapies. We want to make sure that chordoma patients can benefit from these advances. We also want to make sure that drugs that may already exist that could be applied to chordoma are repurposed as quickly as possible. And so a major focus of this campaign is drug repurposing. It's also critical that every day patients are being treated with chordoma, and every day there's an opportunity to learn from that experience. What treatments are working, what treatments aren't working, what correlates with better outcomes or poorer outcomes. And there's a lot that we can do to learn from those patient experiences. And finally, we can't ever lose sight of the fact that it's critical to be there as a source of support and guidance to improve the experience of everyone who's dealing with chordoma, not just in the future, but today and every day going forward. So now I want to dive into some really exciting progress that's been made in each of these 5 strategic priorities, starting with brachyury drug discovery. A quick bit of background for those less familiar with brachyury, and even for those who are familiar with brachyury, the science around this is constantly changing. So I think it might be helpful to quickly set the stage as to what we're talking about when we talk about brachyury. So brachyury exists really in 2 instances. Brachyury is both a gene that is encoded within our DNA, and that gene codes for a protein, also called the brachyury protein. So when we talk about brachyury, sometimes we're talking about the gene and sometimes we're talking about the protein. And it can be important to differentiate because they are relevant in slightly different ways. You may have seen sometimes brachyury denoted as either capital T or TBXT. And that is actually a symbol that geneticists use to denote the brachyury gene. So TBXT, when you see that, that means the brachyury gene. That's not used to refer to the brachyury protein. And so what do we mean when we're talking about the brachyury gene versus the brachyury protein? So the brachyury gene is a stretch of DNA that is in every single cell in every single person's body. We all have the brachyury gene, and ordinarily we all have 2 copies of that gene. The Brachyury gene, or parenthetically, is actually shared among lots and lots and lots of creatures. So basically every vertebrate, and even invertebrates, going all the way back to sea squirts, have the Brachyury gene. So in evolutionary terms, it's the term that's used is it's conserved among different species. And one of the things that that tells us is that it's really, really important for a lot of different biological functions. The Brachyury gene that Chordoma patients have has a slight misspelling in it. So whereas in the general population, there are a number of different varieties of the Brachyury gene. Maybe one has a misspelling here, the other has a misspelling there. Essentially, all Chordoma patients have a particular misspelling in their Brachyury gene, and this is highly predisposing to causing Chordoma. Now, importantly, it's not necessarily causative. So if you have this misspelling, it doesn't mean you're going to get Chordoma, but it means you're at a higher risk. The other way to look at it is if you don't have this misspelling, if you don't have this SNP, you essentially have no risk of getting Chordoma. Another really interesting thing is that very, very rarely there are instances of families in which multiple members are affected by Chordoma. This is called familial Chordoma. And in those families, it's been discovered that they actually, rather than having 2 copies of the Brachyury gene, they actually have an extra copy. They have 3 copies of the Brachyury gene. So these pieces of evidence suggest that Brachyury plays a really important role in the formation or what's called the pathogenesis of the disease. And so what happens? That Brachyury gene gets turned into a protein, and ordinarily that protein is present during development. It plays a really important role in the normal formation of an embryo, but it's supposed to get shut off midway through development, and by the time you're born, it shouldn't be there at all. However, in a number of different cancers, notably in Chordoma, Brachyury gets re-expressed in cancer cells. And in Chordoma, it actually happens essentially all the time. So basically every single Chordoma has high expression of Brachyury. But importantly, it's not present in normal tissue. And that's a really key point that I'll further emphasize here in a moment. So while we've known that for a while, we've known for over a dozen years that Brachyury is present in Chordoma, but not in normal tissue. Over the last dozen years, there's been this accumulation of evidence that has suggested that not only is Brachyury a kind of a unique marker for Chordoma, but also it's a key driver of the disease and it's critical for survival. And not just critical, but last year there was a really fundamental paper that came out showing that it was the most critical gene for Chordoma's survival. So if there was any question before whether this was important, that question has now been answered. Brachyury is the Achilles heel of this disease. And because of those features, there are a couple really exciting and interesting therapeutic opportunities that Brachyury presents. So number one, you can imagine you've got this thing that's uniquely expressed in tumors or in Chordoma, but not expressed in normal tissue. And so could you find a way to target that thing, to target a therapy to cells that are expressing that thing, in this case Brachyury, but not direct them to normal cells? So that's what we mean by targeting expression. Could you somehow kill cells that are expressing Brachyury? The other opportunity is to take advantage of the fact that Brachyury is not just expressed in Chordoma, not just present in Chordoma, but that it's critical for the survival of Chordoma cells, and actually turn it off, and in doing so, kill the cells in which it is expressed, and namely kill Chordoma cells. And because it's not expressed in normal tissue, that presumably would spare or not harm normal tissues. So some of you may be familiar with the fact that there, in fact, over the last number of years have been a number of efforts to try to develop therapies that target the expression of Brachyury. These were initially started not for Chordoma, but for other cancers in which Brachyury is expressed, lung cancer, colon cancer, prostate cancer, for example. All of this work essentially originated at the National Cancer Institute in the tumor immunology group led by Jeff Schlom and Claudia Polena. And they, over a number of years, have developed 3 different therapeutic vaccines based on 3 different technologies. Ultimately, those vaccines were licensed to 3 different companies. I want to apologize here for the formatting in the slide. These question marks are supposed to be arrows, so forgive me. But this is intending to denote that these vaccines were licensed from NCI to a company called Bavarian Nordic, One of them was licensed to a company called Globimune, then on to Celgene, and then on to a company called Nant, and so forth. And so over the years, there's been a number of clinical trials. In fact, I think some of you on this webinar may have even participated in some of those clinical trials. And the exciting news is that there have been some encouraging initial signs of activity. Too soon to say conclusively whether these therapies are working for chordoma patients, but there is some strong evidence that perhaps in combination with radiation therapy, these vaccines could be effective. And there's some additional ongoing trials and planned trials to help really nail that down. However, this is really only one piece of the puzzle. It is the case that as promising as that approach sounds, cancer is a clever adversary. It is constantly evolving, and there's a lot of different ways that it can evade the immune system. There's a lot of things that could go wrong, or I think said differently, there's a lot of things that have to go right for a therapeutic vaccine to work. And so 110% worth pursuing, but we can't put all our eggs in that basket. We have to pursue other approaches that could directly target brachyury and don't rely on a functioning immune response. Now, for those of you who may have been following this for a while, you probably have become accustomed to hearing that brachyury is a difficult drug target. Oftentimes, it's referred to as an undruggable target. And the reason being is that for many, many years, the type of protein the brachyury belongs to, called a transcription factor, that type of protein has been really challenging for drug discovery. There are dozens and dozens and dozens of transcription factors that are implicated in a lot of different diseases and a lot of different cancers. And there have been a long litany of failures in trying to develop drugs against them. And so for the last 15 or 20 years, it's developing drugs against brachyury, against transcription factors has really kind of fallen out of fashion, and industry and academics focused attention elsewhere. However, in the last 4 or 5 years, there's been an emergence of some really powerful new technologies that are starting to overturn that thinking, that are starting to provide an opening to developing drugs against transcription factors like brachyury. And just to go one layer deeper, one of the big reasons why transcription factors have been such difficult drug targets is that they don't have the structure the way that many other easier drug targets, they don't have the structure that enables them, that enables small molecule drugs to easily bind to them. So essentially, they don't have a binding pocket or what's called an active site that a small molecule could easily fit into. And so that has posed a major challenge for drug discovery. However, some of these new technologies provide the possibility of finding alternate ways to get drugs to bind to a protein like brachyury. And so recognizing that potential over the last several years, we've begun investing in kind of laying the ground to bring these technologies to bear on brachyury to make it possible to kind of put brachyury at the front of the line, if you will, for taking advantage of these new technologies and these advances, and hopefully fast tracking the development of a brachyury drug. We began investing in developing some critical tools that would be needed back in 2006 with a grant to Baylor College of Medicine. We collaborated with researchers at the University of Oxford, who in 2017 and 2018 made the extraordinary advance of actually, for the first time, defining or solving the structure of brachyury. So we actually know what it looks like, and we can use computational tools to model it and to do virtual screening of compounds against it. And then many of you have heard me talk about before, we were extraordinarily pleased and fortunate to partner with the Mark Foundation for Cancer Research to fund 3 therapeutic innovation awards back at the end of 2018, with the goal of applying 3 different approaches to develop binders to brachyury. So to find ways to overcome that key hurdle, that key first step that would be needed to enable the discovery of drugs against this target. These were very much high risk, certainly very high reward pilot projects. They were funded for a period of 1 year, and admittedly, they were long shots. We pegged the probability of success simply based upon success in the past in developing drugs against transcription factors as quite low. However, we were just beyond thrilled over the last year to learn, in fact, that all 3 of our grantees have, in fact, succeeded in identifying compounds that bind to brachyury. You've heard me say that before. Some of you have heard me say that before over the last several months. However, this is actually the very first time that we're actually able to visualize it in 3D. So what you're looking at here is a 3D representation of the brachyury protein sitting on DNA. So DNA here is represented as the blue double helix. The brachyury protein here is in gray. And then these stick figures are very, very small compounds that are actually binding to small pockets or grooves within the brachyury protein. So this is something that that for a long time was thought to be highly unlikely, if not impossible. And the extraordinary thing is that this is the toehold. This is the kind of the foot in the door to now being able to develop drugs against this protein. So this is just a zoomed in view of what that looks like. And I mean, I have to pinch myself sometimes. It's just incredible what technology enables, that all of this is happening in in solution in X-ray machines, and and then we're able to visualize using 3D modeling software what is actually happening in 3D space. It's really quite incredible. So kind of where does this put us in terms of developing a drug? So probably it won't come as a surprise to many of you that drug discovery is a long, costly, and arduous process. It takes tens of millions of dollars, and in ordinary circumstances, the better part of a decade to develop a drug. We think that there's ways that we can fast track that and take advantage of the fact that brachyury is an orphan disease to reduce some of those costs and accelerate that timeline. But basically where we are now is we have succeeded in this first stage of screening and discovery. And so we've got a series of hits, essentially compounds that are binding to our protein of interest. Now we have to go through essentially the what is a well-worn path in the drug discovery world of bringing those hits forward through various stages of optimization and ultimately into the clinic. And so I won't go through this in detail, but essentially this lays out the level of funding that's required as well as the potential sources. For the next couple years and several $1 million, this is still an extraordinarily risky endeavor from the standpoint of from a company or an investor. And so it really requires philanthropic dollars to get these promising hits to the point where a company or companies could pick them up. But eventually, if we're able to do that, we shouldn't have to take this all the way to the clinic with philanthropic dollars. Eventually, there will be for-profit companies and investors for whom this becomes an attractive opportunity and can finance later stages of the preclinical and clinical development. Additionally, I think it's important to note that the NIH has a program specifically designed to help rare and neglected diseases, Chordoma fitting squarely within that mandate, bridge what they call this valley of death, this kind of period between where philanthropic or government dollars typically leave off and where industry might pick up. And we've been in very close communication with the TREND program and see that as a golden opportunity to fit into once we have compounds that would qualify for their program. So what I'm excited to talk about now in a bit more detail, and what we announced just recently in our last newsletter, is that building upon the success of the first phase of our partnership with the Mark Foundation, we just in the last several months have awarded a second phase project to an extraordinary team of researchers from Oxford, UNC, and the Institute of Cancer Research, a $1.4 million grant co-funded with the Mark Foundation, as well as an additional $700,000 in research that's going to be done in contract labs. So they could bring essentially the full resources of industry to bear on making this project go as quickly as possible. What does that actually mean? That actually means that there are 6 full-time chemists in India who are working on synthesizing compounds. There's a lab in China that's working on looking at how those compounds interact with the Brachyury protein. So a lot that's happening here in parallel. This is essentially an industrial scale drug discovery project, and it's no accident. Both David Drury and Paul Workman have a long history, 30 plus years or 25 plus years, each of them as drug hunters in industry. There's an interesting personal connection as well. Both David and Paul come to Chordoma and Brachyury research for personal reasons. David, having become friends with a young Chordoma patient through his daughter named Justin Strauss, who many of you may know or knew of, who sadly succumbed to Chordoma, and Paul Workman, who amazingly actually lost his mother to Chordoma quite a number of years ago. So this is both an interesting academic and intellectual exercise, but also quite a personal mission for this group of researchers. So what is this project aiming to do? Several things. So at the end of the 2-year project, the goal is first and foremost to be able to block Brachyury in cells, pure and simple. That's what we need to be able to do. That's what we need to be able to do to get to the next point in the drug discovery cascade. Those compounds that we use that are created need to have properties that enable them to be used as templates for developing drugs. And importantly, I think it's really important to emphasize this. We're not thinking here just about developing drugs for Chordoma. We think that drugs against Brachyury have applicability for many other cancers. However, because compounds have not yet been developed that enable that hypothesis to be tested in the lab, there's a little bit of a catch-22. We don't know for sure whether they're going to be useful in these other diseases until these compounds exist. So the goal is to, even while drug discovery and development are progressing, to be able to have compounds that could be shared with lots of different academic labs so that they could start testing them in models of breast cancer and colon cancer and lung cancer, et cetera. And ideally, if they show promise in those models, that more than anything else is going to create a business case for getting companies and venture capitalists, for example, to invest in the further development of these compounds. So something really important to emphasize here, whereas typically drug discovery is very much a closed and protected endeavor where patents are filed and data is kept very close to the vest for fear of perhaps harming or weakening a patent. This is the exact opposite of that. We reckon that there is a lot that needs to be done that many different labs can benefit from before we need to start filing, before anyone needs to start filing IP. And so if we can create a set of tools and reagents and data, that'll essentially be a tide that raises all ships. And I guess maybe to use another analogy, what we want to do is essentially, in a pre-competitive and completely open way, take the ball from what is now the kind of the 10 to the 20-yard line, take it to the 50-yard line in a completely open way, so that then at that point, companies or other academics can start building upon that work and competing and doing what they need to do from an intellectual property standpoint at the later stages. But in the meantime, just move as quickly as possible and leverage as many different bright minds and resources as we can by virtue of not having to be secretive or protect anything. So all the resulting compounds are going to be freely accessible to the research community, to any academic or company. The data essentially will be shared in real time, and anyone, including any of you, can follow along as this project progresses in a blog that the project team is keeping at this URL, Open Lab Notebooks, and that'll be up on the website as well as in a blog that we just put out earlier this month. So a really different way of doing early drug discovery, but we think it has the opportunity to make this part of the process move a lot more quickly. And importantly, I can't emphasize this enough, hasten the validation of brachyury as a target in a lot of other tumor types, which will create the business incentive that we're going to need to be able to ultimately get these drugs to reach chordoma patients. So a lot happening there, and even more that I don't have an opportunity to talk about now. I'm always happy to take further questions or to talk one-on-one. So please, if there are questions about this, don't hesitate to ask. I want to move on now to immunotherapy, which I think is an area of great opportunity, both for chordoma as well as for other diseases. This is certainly something that probably many of you have heard a lot about in the news and in the media. It's for good reason, because there have been some extraordinary success stories where immunotherapies have been applied to other cancers. And when it works, it tends to really work. There are patients who have legitimately been cured by various immunotherapies, or who have had long-term responses that have been unheard of in certain tumor types like melanoma, which for a long time was essentially a death sentence or incurable, in some instances of lung cancer and kidney cancer, et cetera. So based on those successes, there are now, and this is changing every day, but most recently, as of 2019, over 3,800 immuno-oncology drugs that are in clinical development. That's an extraordinary number. And it's even been actually, people have lamented there are more immunotherapy drugs than could ever actually be tested in cancer patients. So what we need to do, and really the onus is on us as a community and as an organization, to figure out how do we be smart about applying these to chordoma. We can't just go about it willy-nilly. We can't just say, hey, this worked for melanoma, this worked for some other cancer, let's try it in chordoma. That is, we're never going to get through all these 3,800 plus drugs or more if we don't do it systematically. So there's some really important questions we have to answer about how chordoma interacts with the immune system. And I've talked about those in the past, so I won't go into them into great detail, but I do just want to talk about two near-term priorities, two projects that are essentially teed up. So one is we need to identify whether there is a chordoma-specific cell surface protein. Is there something about the outside of the chordoma cell that is different than other normal cells in the body that could be recognized by the immune system and that could be used to direct either an antibody-based therapy or what's called a cell-based therapy? So you may have heard of CAR Ts. These are actually engineered T cells that can be directed to specific receptors that cells have. But the key question is, what is that unique receptor on chordoma? CAR Ts have been developed for a number of blood cancers where those unique receptors or unique proteins have been identified. We don't have that yet for chordoma, and so we have to answer that question. So there's a group of researchers up in Toronto that we've provided some initial support to get started on this, but and they've got some really encouraging initial findings, which are going to be presented at our upcoming research workshop. But that's just the beginning, and pretty soon there's going to be a need to invest further to build upon their work. Additionally, we really need to understand how to make drugs like nivolumab or Keytruda, for example. These are checkpoint inhibitors that you may have heard of, which are being tried in a number of different cancers, including chordoma, how to make them more effective. It seems like they're working for some patients, but not others. So why are they working for some? Why are they not working for others? And what can we do to make them effective in those patients that they're not currently effective for? What can we do to overcome the resistance mechanisms that tumors have in some cases to these therapies? So those are projects that are essentially teed up, and we're currently in the process of raising funds for. In the meantime, we also recognize that the chordoma immunotherapy and understanding how chordoma interacts with the immune system is really in its infancy. And we want to, it's really important that we build expertise and we bring to bear the brightest minds in immunotherapy research on this problem. And so to do that, we've forged a partnership with the Cancer Research Institute. Some of you may be familiar with this partnership. We announced it last year. But the idea is to essentially put chordoma on the radar of the world's leading cancer immunologists to get them focused on chordoma, applying what they know and their technologies and approaches to this disease. Last year, we sourced 18 different concepts along with CRI. 8 of them passed initial review and were invited for full proposals. That review process is ongoing right now. And our aim is to make at least 2 awards by the summer in partnership with CRI. So very excited about this, and we'll certainly keep everyone posted as this unfolds. Finally, I just wanted to share some late-breaking developments in the immunotherapy, on the immunotherapy front. There was a really interesting paper that came out just last month from colleagues, really a group all over the world, but in particular, our colleagues in Vancouver, led by Dr. Torsten Nielsen. And they had this extraordinary interesting finding that they looked at across a lot of different sarcomas, and they were looking in particular at a protein that's expressed on the cell surface called CD47. And this is basically what people colloquially call a don't eat me signal. It basically tells a type of immune cell called a macrophage to stay away. And there have been some, based on the fact that that a lot of cancers express CD47, there have been CD47 antibodies that have been developed that are showing some really significant promise in early clinical trials. But the really interesting thing is that among all the sarcomas that were studied, chordoma had the, all chordomas expressed CD47, which is pretty unusual. And chordomas had the highest level of expression of any sarcoma. So clearly this is telling us something. We don't yet know exactly what. Just because it's there doesn't mean it's playing a critical role, but definitely something that has caught our attention that we are pursuing very actively and rapidly. So as next steps, we are exploring the possibility of testing some of the available CD47 antibodies in chordoma animal models through our drug screening program. And we're working with our medical advisory board to determine whether it might make sense for chordoma patients to participate in some of these early trials, whether they could even be eligible for those trials. So I want to move on now to another area where there's been significant progress, drug repurposing. Just to give a sense of the kind of overall paradigm here, essentially the universe of drugs outside of the immuno-oncology world, these are basically drugs that have been tested in clinical trials, is about 5,500 drugs, and this is growing all the time. Previously, we had tested about 2,500 of those. This was several years back, and you know, that is the goal there is to see whether any of them are able to kill chordoma cells in the lab. Once you do that, you want to be really sure that they not only can kill cells in a petri dish, but they can actually shrink a tumor in a living organism. And it's very, very costly, time-consuming, difficult to get a clinical trial going. Furthermore, you really want to be sure if you're exposing patients to the risk that a drug might pose, that you really have good rationale and strong confidence that that drug is going to work. And so the way to kind of bridge that gap between testing drugs in cells and going into a clinical trial is through testing drugs in mouse models. And one of our real kind of flagship programs and key accomplishments, one of the things I think I'm certainly most excited and proud of, and that is maybe one of the biggest value adds for the research community, is having developed a preclinical drug screening program where now we can rapidly bridge that gap. We can, in the span of a few months, test any drug that's been identified in any lab across the world in a panel of chordoma mouse models and determine whether, in fact, they are able to shrink chordoma tumors. So right at this moment, last year, thanks to the generosity of a donor in our community, the Broad Institute is currently testing all 5,500 drugs in chordoma cells. I'm going to talk a little bit more about how COVID is affecting that later on, but I guess to cut to the punchline, they were able to do a lot of this screening right before labs shut down. So thankfully, that data is now being analyzed and processed. We've got 13 more drugs that are in queue for in vivo testing in animals, and then one more clinical trial that is being planned, plus more that are coming down the pike. One thing to note, as I just mentioned, at the Broad, they were able to screen these the 5,500 compounds in 2 cell lines. That has given them enough initial data to kind of start the computational part of the project, and they're basically doing that now. As soon as operations in the lab resume, they'll test 3 more cell lines and do follow-up validation experiments. In the meantime, in the last several months, we've tested 12 drugs or combinations in mouse models of chordoma. 10 of those recently went up on our website. So one of the great things about this program is because we generate the data, we can make it available immediately without having to wait for it to be published, which can take a year or 2 years or sometimes even more than that. So now in total, there's 94 drug screening experiments that have been put online pre-publication. And when we say drug screening experiment, that means 1 drug tested in 1 model. That's not 94 different drugs, it's 46 drugs. But on average, each drug is tested in anywhere from 2 to 3, or if it shows promise, even more models than that. And this is just a representative example of what some of that data looks like. Quite encouraging. So here you see in black, what you're looking at is the volume of tumors in animals that are untreated. So you could see it's growing consistently. It's actually basically exponential growth. And here you can see in the colored lines how the tumors are growing, or in this case, not growing, when treated with various drugs or combinations. And essentially the takeaway message here is that there's 2 different classes of drugs here that are being tested. One is a class of drugs called CDK4/6 inhibitors. One is called EGFR inhibitors, as well as a combination thereof. And we've known that both of these classes of drugs individually show significant activity in models. What we're trying to see here is whether together there's any synergies, whether they might actually induce not just tumor stasis, but actually regressions. And what you see is that perhaps there is a little bit of added benefit of the combination, but it's probably too small to be statistically significant. So this experiment needs to be repeated with more mice and more models, and probably for a longer time course. But nonetheless, a pretty interesting hypothesis to continue pursuing. I just want to take a moment here while we're talking about drug repurposing to talk about kind of where we are, where clinical trials stand. So a number of these are repurposing trials. Palbociclib, afatinib, pemetrexed, for example. Cetuximab is upcoming. So here you can see there are a number of clinical trials that continue to enroll. One that recently completed enrollment anyway. Cetuximab is upcoming and pending funding, which we hope to raise in the coming months. And then another type of immunotherapy trial that is in early initiation. All right, moving on to the next priority. There's so much has happened. It's hard to cover it all, but I think this is an exciting one. We've for a long time just known how important it is to be able to better characterize what's called the natural history of chordoma. How does it behave? What are the range of different outcomes that patients might experience, especially patients who are kind of fall in different risk buckets or who have had different treatments or different presentations of the disease. And the only way to do that is to run a prospective study to follow patients over time. This is a massive undertaking. It's been on our to-do list basically since the beginning of the foundation, but it requires a lot of expertise to do it right. It requires a lot of resources, dedicated personnel, et cetera. And I'm just extraordinarily happy and grateful to share that through a partnership with the National Cancer Institute, chordoma is now being included in a rare tumors program through the NCI. It's called MyPART, My Pediatric and Adult Rare Tumors Network, where essentially they are going to be following quite a number of patients with various rare cancers, but chordoma is one of about a dozen rare cancers that are now going to be included in this protocol. And And there's some questions that all patients will be asked, but we're working with them right now to develop a specific set of questions and specific set of data that's going to be collected just on chordoma patients to make this relevant, not just to rare cancer, but specifically relevant to the unique challenges and circumstances that chordoma patients face. This is a program that's going to be open to all chordoma patients, but there'll be a particular focus on 2 different subsets, so pediatric and young adult, as well as patients with advanced disease. And there's several important aims of this study. So as I mentioned, to define the range of outcomes that chordoma patients might experience. Importantly, to identify risk factors, so patients can have a better understanding where they might fall relative to the kind of overall risk of the general population. To identify anecdotal responses to therapy. We hear about these through our patient services and through speaking with patients, but which treatments are working, which ones aren't. This might help generate hypotheses about future clinical trials. And importantly, there's a lot that we can learn about how chordoma behaves that might actually help us design better clinical trials in the future. So a lot of important purposes, and a lot of important aims that this particular study will serve. And so if anyone's interested in participating, you can go to this URL or contact the email address on the screen, or contact our patient navigators at support@chordoma.org. And finally, last but not least, I want to talk about some exciting advances in improving the patient experience. So what we've recognized and what we've heard is that over the last 4 or 5 years, we've, I think, come a long way towards addressing a lot of the initial challenges that patients and caregivers faced in terms of getting access to treatment, figuring out what treatment was appropriate for dealing with chordoma, whether it be primary diagnosis, recurrence, et cetera. And there's a lot of material that's been generated on the website. Our patient navigators are really well versed. We have guidelines, just a whole suite of resources. However, what we've heard and what we've observed is that what's lacking now, where there's a gap, is dealing with all the issues that come after treatment, the side effects, the ongoing issues that, quality of life issues, rehab, recovery, physical therapy, et cetera, emotional challenges, changes to one's routine. The whole gamut of survivorship issues remains one that we've not been able to fully or adequately address. And so that's an area of priority and a big reason, or a big focus of ours going forward. As some of you may have seen, late last year, we released 3 videos in our Expert Answer series focused on survivorship questions. We also are just about to launch, I'm very happy to say, a new specialist directory within Chordoma Connections. So many of you may be familiar with our doctor directory that's focused on surgeons, medical oncologists, radiation oncologists. However, we know that, and in that case, there's clear criteria that our medical advisory board has established for membership in the doctor directory. However, there's a whole constellation, a whole team of other specialists that we know are really important for managing the full spectrum of survivorship needs of patients. Is everything from physiatrists to ENTs to pain specialists to psychologists, et cetera. And it's unlikely that these doctors are going to necessarily have published on chordoma. It's unlikely that many of them will have seen huge numbers of patients, but members of our community will have had experiences with some of them, and those experiences are valuable to share. And maybe you've had a good experience with a doctor that might help make it easier for someone else to find a specialist near them. And so just as we've got this doctor directory for the kind of core specialists that are involved in the primary management of chordoma, we're creating an internal specialist directory in Connections that'll be live soon, that importantly anyone can contribute to. So we encourage you to check that out. We'll be announcing that here in the next couple weeks and months. And then also, just to put on everyone's radar, we have a new survivorship survey that we're launching in the next couple weeks, or the next few weeks, that aims to more fully understand, help us more fully understand the full range of needs and challenges that the community is facing, and therefore help tailor our services going forward. So I know that was a lot. I'm going to now pivot towards talking about what we are doing to continue our mission in response to COVID. And I'll break this down basically in each area of the organization. And I'm not going to belabor these points because I think the take-home message is that, yes, there are challenges, but we're finding ways around them. And we are really well positioned at the moment to continue fulfilling and moving the mission forward. So out of the research labs that we're currently supporting, most of them right now are in a work from home situation. However, at least in the near term, they've been able to pivot towards computational and data analysis work. So maybe the project timeline has shifted, but hasn't necessarily extended dramatically. If the lockdown extends a lot longer, if we have to go back into lockdown, that could change. But for now, there hasn't been a massive disruption to overall research timelines. In particular, the Brachyury drug discovery project is going at essentially nearly full speed, thanks to the labs that are doing a lot of the work in China and India. And then our drug screening program is essentially at full capacity. In fact, we're doing more experiments now than we've ever done in the past. And hats off to our partners at START down in Texas for just doing an extraordinary job of keeping operations going. To get mice, to get a colony of mice up and running takes months, if not years. And so the fact that that hasn't gone down is just a real godsend. We're also proactively exploring additional outsourcing options to avoid delays if the lockdown continues or if it's reimposed. One area that has been put on pause is the tumor donation program, or our biobank, as you may have heard it called. And then importantly, our research workshop, which was scheduled for this summer, has been turned into a virtual workshop and is currently being planned. But we are planning to reschedule, or we have rescheduled the in-person workshop for the summer of 2021. We've definitely had some questions during registration and from quite a number of people who have contacted us about the status of clinical trials and how they're being affected by COVID. So the short story is most are still enrolling. There's some site-by-site variation. I won't go through the details here, but basically, if you have any questions about whether a trial is enrolling, best to contact the trial site or contact our patient navigators at support@chordoma.org, and we can tell you the latest as to where each site is actually in terms of enrollment. From a patient services standpoint, patient services are operational. Shannon and our almost brand new patient navigator, Andrea Locke, who joined us earlier this year, are working 7 days a week and available to answer questions that come in, and always just a phone call or an email away. As I mentioned, we're going to have a virtual Ask the Experts this summer, so please stay tuned for that. And then we really hope to see everyone in person in 2021 in Boston for the rescheduled community conference. And you'll be hearing more from us about that in the months ahead. So from an operations and finance standpoint, clearly, as you can see from my background here, we're all working from home. We already were largely set up to work remotely, but we have really now shifted 100% of our operations to being virtual. This has been actually a surprisingly seamless process, and hats off to our Director of Operations, Tammy, and really the entire staff for rolling with the punches here and not missing a beat despite the challenging circumstances. While for the most part we're weathering this quite well, and thankfully thus far we haven't seen a significant fall off in donations, and really I can't underscore how thankful we are for that. We do know that there are kind of storm clouds on the horizon and a lot of economic uncertainty, certainly fundraising events that have been or will be canceled. And so we're projecting a pretty significant impact on revenue in the coming months, and are needing to adapt to make sure that we're able to extend our runway and continue operations despite that shortfall. I can't thank everyone who has contributed in recent years enough and as well as our board for really the careful financial stewardship and financial planning that has put us in a pretty good financial position to weather this storm, at least for a short period of time. I think that by and large we're in a better position than many of our peers, and for that I'm just extraordinarily grateful. We're also very grateful that we applied for, despite a lot of challenges, but we did actually apply for and receive a loan through the Paycheck Protection Program, which essentially will cover 2 months of payroll. We believe that this loan will be forgiven. In other words, it will turn into a grant at the end of the 2 months, but that is still very much up in the air, and there's some uncertainty around that. And then from a fundraising standpoint, not surprisingly, a lot of the in-person events have been postponed or canceled. However, I want to thank everyone who has continued to contribute online, who has continued to organize through your champions pages. This has really been the lifeblood for us in the last couple months here, and we are just eternally grateful. I mean, contributions are always valuable, but especially valuable now, and so really want to thank everyone who has continued contributing and has continued raising funds within your network. I also want to share that our new head of philanthropy, Kenny Brighton, who joined our team late last year, is here as a resource to help if there's anything that anyone needs from an assistance standpoint, either in doing an online event, in approaching another foundation, approaching a donor, a philanthropist, or discussing a gift of your own. Kenny is here and eager to be of service. So please don't hesitate to reach out to Kenny or to me for that matter. We're all at first name at Chordoma.org. So Josh at Chordoma.org or Kenny at Chordoma.org. All right, so recognizing that we're a little over here, I'm gonna focus now on current priorities and run through this relatively quickly. So I think first and foremost, I want to just say that above all, our current priority is being there to meet the needs of our community, however that might evolve in response to COVID. So whether it's finding appropriate care, whether it's dealing with delays in care, emotional challenges, whatever it may be, we want to hear from you and we're here to help. So please don't hesitate to reach out, again, to our patient navigators, or really to any of us. We also, first and foremost, need to be there for our grantees and our collaborators, and are doing everything we can to stay in touch with them and to help them problem solve and troubleshoot through the challenges that they're facing with COVID. But we also have to continue looking forward and continue raising funds to advance our priorities and continue the momentum that we have on the campaign. So this means continuing to raise funds to support our operations, our patient services, and in particular, our immunotherapy initiatives, as well as upcoming clinical trials, particularly the cetuximab trial. From a kind of new project and new initiative standpoint, some really exciting things on the horizon that we're working on, even in the midst of all that's going on. I won't go into it in detail, but we're working on creating centralized repositories of all those materials and tools that I mentioned that are so critical for brachyury drug discovery. We've got a number of pretty exciting investments that are planned in immunotherapy that I've touched on throughout the webinar, and are just really excited and eager to move forward on those. From a drug repurposing standpoint, Joan is actively working on testing, working through the queue of drugs that we have for in vivo testing. So there's 9 new drugs that we're working on testing, and then 4 drugs that have previously been tested in some number of models and have shown promise, but now need to be tested in additional models. And then, as I mentioned, the upcoming cetuximab trial, as well as the survivorship initiative that we've talked about. So that essentially concludes the presentation part of the webinar. I want to thank you for your interest and your attention. As you can tell, there is a lot that's happened. The mission continues to move forward despite the challenges that COVID is posing. We remain here as a source of support for the Chordoma community, for the research community, for physicians. And, I think first and foremost, we're in this together. So if there's anything that we can do to be helpful, if you have any needs, any questions, don't hesitate to reach out. I'm now going to open it up to questions from the group and bring on my colleagues, Shannon Lozinsky and Joan Levy. I will also mention that while we have this webinar scheduled for 90 minutes, and I don't expect everyone to stay on for more than 90 minutes, we definitely can. And so if we need to go a few minutes longer to answer questions, we can definitely do that. So without further ado, I'm going to stop my screen share and welcome on Joan and Shannon. All right, let's see if I can get them unmuted here. Hi, Joan. Hi, Shannon.
Hello.
Hello, Josh and everyone.
Okay, so we've got a number of questions that have come in. So we have let's see here. We have a question from Sharon Berlan. How does the COVID shutdown affect the development in trials or in the lab? Is progress still happening? So I think we've addressed that to some extent, but maybe, Joan, I could direct that to you from the standpoint of new trial development and the pipeline of drugs that are making their way into new trials. Do you want to expound on that just a little bit?
Yeah, sure, Josh. I'm happy to do that. So as Josh mentioned, our drug screening program that we're very proud of is fully functional, and nothing has been slowed down. And we are testing a number of different promising drugs in animal models through this program. We've done a number of experiments, tested over 10 drugs just in this, the last 6 months, some with encouraging results. And what we're going to do, those that look encouraging, we then go to vet them in front of our medical and scientific advisory boards to see, to get their advice as to whether they should go forward into clinical trials. And if they give the thumbs up, we then work with the investigators and companies to advance these drugs into clinical trials. So again, we've gotten some encouraging results recently. We're going to test them in more models, put together the data, and hopefully advance them into trials for all of you.
Thanks, Joan. And just to add to that, I mean, one of the things that has been eye-opening to me over the last number of years is just how many steps there are before from idea or promising result in a lab to actually getting a clinical trial initiated. And typically this takes anywhere from 1 to 2, if not more, years. We're trying to cut down on that timeline, but most of that work does not involve being in person. Most of that is the investigator or the company designing the protocol, raising the funding, getting ethics boards to approve the protocol, getting companies to authorize release of the drug. So there's a lot of kind of behind the scenes hoops that have to be jumped through that frankly are, should not be affected, I wouldn't say, by, and have not thus far been affected by labs being shut. So in fact, Joan's actively working with a group of researchers right now who are approaching some companies, and those conversations seem to be going pretty well. Okay, so another question we have is, so people have heard about CRISPR-Cas9. How is that being employed, and specifically, is there anything that's being done with CRISPR to target the Brachyury gene? So Joan, hand that one off to you as well.
Okay, so I'll take that in 2 different ways. We actually were able to identify Brachyury as the major vulnerability by doing CRISPR analysis in research laboratories in different cell lines. So knocking it out showed that it was the most major vulnerability in chordoma. If the question is directed at, can we use CRISPR in the clinic to knock out Brachyury, we don't know yet. We will watch the advances that are being made of using CRISPR technology to edit genes and be useful in the clinic. And we'll follow that very carefully and see at one point if we potentially could apply it to brachyury. Absolutely. But we could think optimistically about it.
Yeah, but definitely something that we're keeping our eye on and that probably holds promise in the future. Okay, let's see here. Oh, these questions are coming in fast now. Okay, so question is, for patients who are needing care in the midst of with a Chordoma expert in the midst of the pandemic, what would we recommend? Shannon, I'll turn that one over to you.
Sure, thanks. So the question of travel to get to an expert is sort of individual of dependent on where you are, where you need to get to, and that kind of thing. But we do know that people are finding ways to travel if that's necessary. As far as getting opinions from Chordoma experts, so many of the doctors in our doctor directory currently had kind of always provided remote consultations and had a great process for that, so that people could get the process going with them before having to travel. And that's kind of expanded during the pandemic, so that even more doctors are doing it, or they're being very adaptive in arranging plans and doing telemedicine visits and things like that. And we do know that that Chordoma surgeries mostly are not being considered elective surgeries. They are being considered necessary surgeries. So if there are still restrictions in place to hold off on elective surgeries, that should not affect a Chordoma surgery. There may be things you have to do to get it approved as medically necessary. They may do tests, COVID tests the day before to make sure that you're healthy, that kind of thing. But it has been happening. It's just a little bit of a adapted process, but it's certainly possible. If anyone has any questions about that, need some specific advice, or if there's anything we can do to help more specifically with your situation, just reach out to our patient navigator, Andrea, at support@chordoma.org, and we'll be glad to help.
Thanks, Shannon. It's been interesting to see kind of how variable it's been. I mean, even among hospitals in New York, for example, which I think arguably has been hardest hit, or for example, in Europe during the earlier wave. So it's kind of a constantly evolving situation, as I think is often said these days, and we're doing our best to keep up. You know, one thing I didn't mention that I really should have, and I apologize for this, but Shannon and Andrea put together just a tremendous resource on our website, where basically they've linked to, well, aside from providing a lot of useful information about COVID and potential impacts on Chordoma patients, they've linked to essentially all of the top treatment centers in North America and Europe that treat Chordoma patients, and their kind of COVID information page that will provide, in most cases, a status update as to which patients are being seen, what precautions are being taken, et cetera. And so if you have further questions about that and you haven't already checked out that page, it's probably worthwhile. It's on our homepage right now. So if you go to the website, you should be able to easily access it. And certainly if anyone, the other thing I'll say on that point is that if anyone hears of any particular restrictions or limitations on getting care at a particular medical center that you feel like could be valuable for other patients to know, definitely let us know. Reach out to Shannon, reach out to Andrea, or put it on Chordoma Connections, because I think we could probably all learn from each other and kind of share some communal knowledge here. Okay, we have a question that's just come in. Why are these new brachyury treatments called vaccines? When I think of a vaccine, I think of a treatment that builds immunity through the production of antibodies. Is that how these treatments are working? Also, why is radiation paired with these treatments? That's such a good question. So, yeah, absolutely right. We think primarily of vaccines basically as being preventative, or what's called prophylactic. In other words, you get a vaccine to develop immunity prior to being exposed to some pathogen. However, in cancer, vaccines can be used in a therapeutic approach as opposed to a prophylactic approach. So in that case, what the vaccine is doing is essentially mounting or triggering an immune response against, in this case, the tumor. And the way that it does that is it presents the immune system with a pathogen that it really hates, either yeast or a virus. And this yeast or virus is engineered to contain within it the target of interest that is on the tumor cells that you want the immune system to go after, in this case, brachyury. So that's why it's called, for example, the brachyury yeast vaccine, because essentially it's yeast that's engineered to express little bits of brachyury. And when the immune system sees the yeast, it goes into essentially overdrive, and it directs a lot of resources to trying to kind of shut that down. And then not only does it do it locally at the site of the injection, but then it sends out what essentially amount to kind of sentries or scouts across the body to look for other signs of what it thinks is an infection or a pathogen. And so when it sees a cell that expresses brachyury, it says, "Hey, this is not good. Let's get rid of it." And it then mounts a response that kills that cell. There's a really important nuance here, which is that brachyury is ordinarily, as I mentioned earlier, a transcription factor, where it resides not on the surface of the cell where it would be visible to the immune system, but rather in the cell's nucleus. And so what the immune system is actually seeing is not brachyury itself in its entirety, but little short segments of the brachyury protein that are presented on the surface of cells by what's called the antigen presentation system. And this is how cells basically communicate with the immune system, and how the immune system tells self versus foreign bodies. So it's a pretty incredible technology. As far as how radiation works with it, there's two thoughts on that. So it's thought that radiation can make cells more susceptible to being attacked by the immune system, but also can make cells more visible to the immune system or more accessible to the immune system. So it might be a 1-2 punch, or maybe even there's more mechanisms at play. There's a lot of hypotheses around that, but I think it's an area of very active ongoing research as to why and all the different ways that immunotherapy seems to be synergistic with radiotherapy. And that's not unique to these brachyury vaccines. That's across lots of different types of immunotherapy and radiotherapy. Okay, we have a question. Your statements around the virtual screening of existing drugs against the brachyury protein seem similar to articles I'm reading about COVID-19 treatments. Obviously, a huge motivation to find COVID-19 drugs. And I suspect these efforts may lead to advancements in underlying science and methods. Do you think there is the potential for advances made in this arena to benefit Chordoma research? Really good question. So, in fact, the library of compounds that is being screened against the Chordoma cells right now at the Broad, it's called the Broad Repurposing Hub. That same library is actually one of the ones that's being used for screening against for screening against COVID-19. I associate CRISPR with the Broad because they did a CRISPR screen for us. So apologies for the lapse there. But yes, in these particular cases, that screening, it's actually not a virtual screen. It's a it's a robotic wet lab screen. And so in that case, it's technology that's been developed in the last several years being applied to both chordoma and to COVID. From a virtual screening standpoint, I think it's the same thing. There have been a lot of tremendous advances in virtual screening technologies in the last several years, like to the point now where you were able to screen billions of compounds virtually and then synthesize them on demand in a way that's never been done before. So that's being applied to chordoma. It's also being applied to COVID. I think at the moment, it's fair to say that basically we're both benefiting from what is truly the cutting edge of screening technologies, both virtual or in vitro. As far as new technologies that are going to be developed specifically for COVID, I think it's probably too early to say whether they will be applicable to chordoma. However, what I can say for sure is that we are constantly scanning and monitoring the kind of full range of screening technologies and approaches, as is our scientific advisory board. And certainly, if there is anything kind of new or extraordinary that comes about as a result of COVID, we're going to be eager to try to apply that to chordoma just as soon as possible. Okay, we have a question about the survivorship initiative. We're right up against the end of time here, so I'm going to take maybe one or two more questions. We have a question about the survivorship initiative and the new directory, the new doctor directory that's upcoming, and how are we going to ensure quality control around that? So Shannon, maybe you could address that quickly.
I can. Thank you. That's a good question. So we, as Josh mentioned, we have a doctor directory on our public main website, and there are inclusion criteria for that were developed by our medical advisory board a few years ago. Those criteria apply to surgeons, radiation oncologists, and medical oncologists only. And we, along with the medical advisory board, determined that for all of the many other types of specialists that may treat survivorship and quality of life needs, that it would be nearly impossible to come up with similar criteria for all of those. Endocrinologists, neurologists, physical therapy, acupuncture, so many different things that people might need treatment for, care around outside of the main treatments for chordoma, that we hear time and again at our conferences that it's hard to find those people. If people have information that they'd like to share about who they've seen and things like that, they would like to be able to do that in some way. So within Chordoma Connections, it's available to be crowdsourced because it's a membership. You have to be approved to be a member. We can keep it in there and allow it to be crowdsourced with the clear caveat that we are not, these doctors have not been vetted by us in any way. We do feel like if you've seen an endocrinologist locally in your area, when you might have been treated at Mass General, but you don't live near Boston, if you found an endocrinologist near you and there happens to be someone else that's their similar, their home area as well, that needs an endocrinologist, that this is at least a way to know that this doctor has treated me well, understands my needs and my symptoms, has taken time to understand chordoma, and I feel really good about them, that, it's one step that we can take to help people connect. But it is necessary to understand that we have not said specifically that we know about their background as far as treating chordoma, and we're not endorsing them. We're not any of those things. And I think that, we want people to understand that, but we want this to be a really helpful resource to people as well.
Excellent. Okay, so just a couple quick final questions. We have a question about how to make a tax deductible donation from Canada, and I would just, on that note, encourage anyone who's interested to go to our website. We have a page on the website for international donations. We also have a affiliate organization there, the Canadian Chordoma Network, and our ambassador, Stephen Golick, who I'm sure would be delighted to speak with you. So feel free to contact us at support@chordoma.org or or go to the website. There are a couple of medical, kind of individual medical questions on here, which Shannon will follow up with folks individually about. And then finally, there's a question about, will this webinar be available broadly? And the answer is yes. It's going to go up on the website. It'll go in our upcoming newsletter, and anyone who receives our newsletter, including donors, patients, people who have contributed to the biobank, et cetera, will get access to it. So I'll leave it there. I just want to thank everyone who joined us today. I really appreciate your interest, your attention. Most importantly, I appreciate your support and your dedication to this cause and to the organization. We would not be here without you, and we could not do what we do without you. So huge, huge thanks for blowing wind in our sails, especially during this difficult time. Just you being here really means a lot to us. And frankly, the outpouring of interest and the amount of participation we had today really even exceeded our expectations. So it feels really good to connect with everyone, even if just virtually, and to know that there is a large group of people out there who care about this work and care about what we're doing. And we are, as I said, we're in this together. And I think that on the other end of this COVID situation, we're going to come out stronger. So thank you for that. And finally, I'll just reiterate that we really want to be better, get better in the future as we do this. I think that hopefully this is the first of several upcoming webinars, both from a general impact and update perspective, but also kind of more topic specific webinars. So stay tuned for that. But please do send feedback, whether whether things that you liked or whether things that you could change, and don't hold back. We have thick skin. The only way we're going to learn and get better is to hear honest feedback. So really, if you do nothing else after this, if you wouldn't mind, if there is something that you want to share, just don't hesitate to share it. All right, signing off. Have a great day, everyone. Hope to be in touch soon. Take care.
Take care. Thank you for joining.